Ah, here we go.
Hello.
Yo, yo.
Hello.
All right, I know you can hear me, so let me go ahead and get started. Are there any questions before we begin? I got a number of assignments graded, including Lab 3 and Lab 4. Lab 3 was supposed to have been graded last week. Sorry about that. But now it's graded. And Lab 4, they can be corrected until this Saturday. Now, I'm not sure when I'll get to Lab 5. So I'm not sure when that one will have to be turned in, but it depends on when I get it graded. Any questions about any of that? Let me see. I guess I should leave this here because I'll need it for the lab. I think there's something I need to talk about with class.
So,
I've never looked at that, so let's take a look to see if anyone's saying anything. Great.
oh, no, not me. Don't read mine.
What?
No, I'm just kidding.
Okay.
Keep going. You can keep reading.
All right. Let's go ahead and get to the schedule. So today is August 1st. I've got to pay my rent. We're supposed to be working on chapter 4 and finishing chapter 4 today and starting chapter 5. Please note there will be a quiz 2 on Wednesday. I haven't finished all the questions, but I hope to get that done tonight. And quiz 2 will cover labs 3 and 4 and chapter 3 and the end of chapter 2. Any question about any of that? Now, in the lab today, we're going to be discussing and
Actually, I have
starting.
a question.
Yeah,
Could you please say where the end of chapter 2 starts?
chapter 2 will start, the end of chapter 2 will start where we started the lecture on, what was that? July 20th. So on Thursday, we covered chapter 2. Is that good enough, or do you want me to say exactly?
Wait. So this chapter 2 refer to your chapter 2, not the book's chapter 2. So like when we do if we stopped in the middle of chapter 2, that would quote unquote be the start of chapter 2. And then when we resume chapter 2, that would be the end of chapter 2.
Yes, although I thought the textbook and my chapter 2 were the same.
They are just when you stopped lecturing, I guess it was like, well, when does chapter 2 begin? But since it's not like on the page, it's going to be more of where I guess you continue.
Let me see if I can answer that a little more specifically. Let me stop sharing my screen and go into quiz 2, I think. Let me look on that. Yeah. So for chapter 2, you do need to know the different of the 4 biological molecules. You should know how RNA differs from DNA. You should know you should know about hydrolysis reactions and condensation synthesis reactions. All right, that's it for chapter 2. Does that help?
Thank you. Yeah.
All right. Let me share my screen. All right, let's open up chapter 4. And we had talked about group translocation, a form of active transport that is only seen in prokaryotes. So you should know the cytoplasm is the substance inside the cell membrane of a prokaryotic cell. It's mostly water, about 80%. It does have dissolved molecules in it, so we call those solutes, including DNA. DNA most of the time is dissolved in the cytoplasm, so you can't distinguish it from the cytoplasm. It does include the nucleoid region. The ribosomes, which are not dissolved in the cytoplasm, they're big enough they do not go into solution. And then inclusions are found within the cytoplasm. Let's talk a little bit more about the nucleoid area. This is the area where the DNA is. It does tend to stain differently when we stain a cell, but there is no distinction between where the DNA is and where the rest of the cytoplasm is, because there's no boundary between them. Wherever the DNA is floating in the cytoplasm, that will be where the nuclear area is that we call the nucleoid region. And with a light microscope, you can't see it, but if you stain it, it does stain a little differently than the rest of the cytoplasm. And there you can see it, the nuclear area in an electron microscopic image. The ribosomes are big enough that they do not go in solution in the cytoplasm, and they would be these little granules here. Let me blow this up a little. Right there. And you can actually see them over here in an electron microscopic image, little dots in here. There's a whole bunch of them right there. Shrink that. The ribosomes are the site of protein synthesis. You don't need to know the different subunits. You should know that the ribosome is composed of two subunits that come together, but you don't need to know the size. And they differ for prokaryote ribosomes and eukaryote ribosomes as well. But you should know the prokaryote ribosome is a little smaller than the eukaryote ribosome, and we say its size is 70S. The S stands for the sedimentation coefficient, where the ribosome sediments in an ultracentrifuge gradient. Any question about any of that? That just explains what 70S means. And that ribosomes are made up of both protein, you can call them ribosomal proteins, and ribosomal RNA that we call rRNA. Any question about any of that? So this right there is made up both of protein and RNA, rRNA. Inclusions are reserve deposits within the cytoplasm where so many molecules of this reserve deposit exist that the deposits start to precipitate out of solution. And when they do precipitate out of solution, we then call it an inclusion. Let's see if I can see an example of that. Oh, there's one right there. Probably that big one right there. Where the cell is just storing so many molecules of one substance that they come together and then they precipitate out of solution. And they're called inclusions. There are different examples. You don't need to know the different examples or the names. You should simply know that there are different inclusions. Okay. Here we're looking at some different inclusions, magnetosomes, and how they look different than sulfur granules. This organism is a sulfur metabolizing organism, and so it accumulates sulfur in its cytoplasm, and so much sulfur forms in one region of the cytoplasm that the sulfur precipitates out of solution, forming the inclusions, which we're seeing here. And then these are metachromatic granules. Let me see if I can blow that up. The yellow lines indicate a cell, and you can see that this cell has 3 inclusions in it. Any question about any of that? All right, the next topic I want to speak of are endospores. Endospores are special survival structures found in only a few bacterial species. When the environment starts deteriorating, the parental cell begins forming an endospore, assuming that it is a species that can form endospores. Most of the species that form endospores belong to the families Bacillus or the family Clostridium. Okay, there are, I believe, 2 other families that also form endospores. However, in one of the families, there's only one member in that family, one species, and it's actually a cockeye. That's why I know that one. And then the other is something specific, like a few sulfur metabolizing bacteria, I think they are. They're not in the family Bacillus or Clostridium, but all the other endospores forming species are in these 2 families, Bacillus and Clostridium. And when the environment deteriorates, the parent cell starts forming the endospore. Here we see, let me see if I can blow that up even further. A parent cell right here in dark purple, and it's forming the endospore, which is that round circular type or ovoid type thing in the middle of this one. And here we see the endospore. Sorry, wrong way. There's the parent cell there, and it's forming an endospore inside of it. It's forming the endospore because the parent cell realizes that the environment is deteriorating, and the environment is pretty soon going to be so poor that the parent cell is going to die. You should know that endospores survive many different environments. They're highly resistant to desiccation, meaning they don't easily dry out. They're resistant to heat, chemicals, radiation. They can survive conditions where the vegetative parent cell cannot. When the environment begins to deteriorate, we have sporogenesis, which is where the endospore begins to be formed inside the parent cell. And then that endospore survives the harsh environment. And then when the environment changes and then becomes good for the species to grow, the endospore will germinate and return to the vegetative state, meaning become a cell without an endospore. And in the vegetative state, the cell can undergo metabolism, can undergo growth and reproduction. In the endospore form, the cell does not grow, does not reproduce, and its metabolism is shut way, way down. It essentially has no metabolism, but it isn't quite none. It's very low in its metabolic activity. Any question about any of that? All right, the first thing you should know about endospores are they are not reproduction. One parent cell forms one endospore, and then the parent cell dies when the environment deteriorates. The endospore continues to survive. When endospore forming is made, the cell undergoes division in the normal way where the DNA will replicate, and then one copy of the DNA will move to one side of the cell, the other copy of the DNA will move to the other side of the cell. So that's all normal. What is not normal is the cell does not divide in half by binary fission. It divides where the endospore will form a smaller part of the cell than the remaining portion, which becomes the parent, continues the parental cell. It doesn't go through cell division. It just continues as the parent cell. And then the spore septum begins to divide. So this will become a separate cell. And then the two membranes are put around the endospore. And you can see the endospore in this case actually moves a little bit. So it moved from this end of the cell to, it's still on this end of the cell, but it just moved over a little bit. And then the cell wall is put down between the two membranes. And then the spore coat forms on the outside of the endospore. And then the environment becomes so disfavorable for growth that the parent cell will then die. And then when that happens, the endospore is released. Let me see if I've got some pictures of that. Here we go. Oops, didn't mean to move that. Blow that up now. Oh, crud. So that's an endospore that has been released. This one is about to be released because the parent cell has pretty much died around it. And then there's an endospore inside a parent cell. So either it is being made or it is about to be released. Any question on endospores? The main thing to know about endospores is they're a survival structure. They are not a reproductive structure, because one parent cell only makes one endospore. All right, that's it for, oops, too small. The structures of a prokaryotic cell. I'm not going to cover the structures of a eukaryotic cell other than say that this slide is showing you or trying to show you the structures you can find in an animal cell on the right and the structures you can find on a plant cell on the left. Most of the structures are found in both, like the mitochondria. There it is, right there and there, can be found in both plant and animal cells. The nucleus found in both, the Golgi apparatus found in both. There are a few which are only found in plant cells, like the chloroplast, only found in plant cells, not in animal cells. And then it has a vacuole here. That should be a, oh geez, let me think about that now. The central vacuole is only found in plant cells. It's actually the largest organelle in a plant cell. You can find vacuoles in an animal cell. There aren't really any shown in this picture, but you can find a vacuole in a animal cell. But the central vacuole is not seen. The largest organelle in an animal cell is the nucleus. You have both membrane-bound organelles, like the mitochondria, the chloroplast, which are separated from the cytoplasm by a membrane. And then you have non-membrane bound organelles, such as the centriole here. Which it is a cell structure, but it does not have a membrane around it. Any question about any of that? All right. Oh, let me briefly talk about this. And that is, if a eukaryotic cell moves, it can move from 3 different ways. It can move if it has a flagellum or flagella, the same as a prokaryotic cell, although the proteins are a little different, but they are still made of flagellum proteins. Eukaryotic cells can move by cilia, which are shorter than the flagellum, and they look like hair-like structures, and they beat in unison to propel the cell. So if it beats this way, the cell will move that way. Prokaryotic cells do not have cilia. And then they can move by pseudopods, where the cell can put out its cytoplasm on this side and pull up its cytoplasm on that side, and that will make the cell move that way. Those are the 3 ways that eukaryotic cells can move. Prokaryotic cells can only move by flagella. Your textbook might not state it quite correctly. I have to check on the latest version of the textbook, whether they got this correct or not. And that is both eukaryotes and prokaryotes have a cytoskeleton. The difference is the cytoskeleton in eukaryotes is very complex. It makes the structural framework throughout the entire cell, making the backbone of the cell. So this is one cell, and wherever that is, we have the cytoskeleton inside of it. Here's another cell showing you different cytoskeletal elements, that blue being the nucleus, but the cell has membranes around here. And the point is, eukaryotes have a much more complex and much more developed cytoskeleton. Prokaryotes do have a primitive cytoskeleton, and this was not known 20 years back, but it was discovered something like 20 years ago. And your textbook author was a little slow on updating the textbook, stating that prokaryotes do have a primitive and simple cytoskeleton, but they do have a cytoskeleton, okay? It's just not as complex or intense as the eukaryote cytoskeleton. You should know that the eukaryote ribosomes are larger than a prokaryote ribosome. They're 80s in size, so they sediment lower down in the ultracentrifuge gradient. Let me state that eukaryotes have an endomembrane system where something can move around in the cell. And it usually starts in the endoplasmic reticulum, making a protein or something like that. And then that endoplasmic reticulum makes a bud, or something buds off the endoplasmic reticulum, which then moves into cytoplasm, and we call that a vesicle, a membrane-bound organelle that moves molecules, in this case proteins, from one location to another. And the molecules move from the endoplasmic reticulum, and really the rough endoplasmic reticulum, because I said proteins, and then they move to the Golgi apparatus. The endoplasmic reticulum actually fuses to the Golgi apparatus, and then the Golgi apparatus can modify those proteins and sort them, and then package them in a bud that buds off of the Golgi apparatus. And when it buds off, it then moves as a vesicle to carry the proteins to where they need to be. It could, for example, fuse with the cell membrane and then excrete those proteins from the cell, like in a teenager with raging hormones. The hormones will be made in the cell and then excreted into the blood. Okay? Any question about any of that? If the proteins are needed elsewhere in the cell, the vesicle will move to where the proteins are needed. If the cell, on the other hand, just wants to store those proteins, when the vesicle stops moving, we stop calling it a vesicle, because a vesicle is a transport organelle. And then the vesicle just stores the proteins, or whatever it's storing, in that location. So the vesicle doesn't move, and now we don't call it a vesicle, we call it a vacuole. So a vesicle, while it is moving, is called the vesicle. And then once it stops moving and starts storing its contents, we call it a vacuole. And that's why animal cells can have vacuoles. Any question about any of that? The point is that eukaryotic cells have this endomembrane system. The protein can be made in the endoplasmic reticulum, moved to a vesicle to the Golgi apparatus, modified in the Golgi apparatus, and that can change the protein, like slice off some of the early amino acids, like all proteins that are made with AUG, which codes for a methionine. If the protein doesn't want methionine, you have to remove that methionine. And that happens in the Golgi apparatus. You can also methylate the amino acids in the protein. and that can be modifying the protein. And that happens in the Golgi, and then the Golgi bends it off in another vesicle, which carries it elsewhere in the cell. That's the endomembrane system, shown here. And the rough ER making a vesicle, making the proteins, stored in a vesicle, transported to the Golgi apparatus, sorted, modified. When that's done with the protein, that protein is then butted off in a vesicle, which carries it to where the protein should be. Now, there's one more topic I need to talk about, and that is the endosymbiotic theory. This is a theory to explain why eukaryotic cells have mitochondria and some eukaryotes have chloroplasts. Plant cells have both chloroplasts and mitochondria, assuming that they're a photosynthetic plant cell. Like the root cells do not have chloroplasts. Any question about any of that? Well, why do we need to explain the evolution of the mitochondria and the chloroplast? These organelles differ from other organelles. For example, the mitochondria and the chloroplast have a molecule of DNA in them. They also, those molecules code for different proteins and structures in the cell, like the ribosomes of the mitochondria are made from the mitochondrial DNA, and they differ from the ribosomes made from the chromosome of the nucleus of the eukaryotic cell. Any question about any of that? So they differ in that these two organelles have DNA molecule in them, and they have a ribosomes. The ribosomes differ from the eukaryote ribosomes. The ribosomes of the mitochondria in the chloroplast are 70S in size. The ribosomes from the eukaryote chromosome are 80S in size. So how did these organisms originate? Let me get paint up. There it is. I blow this up. Well, a long time ago, we had a proto-eukaryote cell, which wasn't a eukaryote cell yet because it didn't have chloroplasts and mitochondria. So it was before the eukaryote cells were around. And we're going to call that a proto-eukaryote. Am I spelling that right? And it engulfed another cell, which I'm going to draw. Oops, I don't have that drawn. Let me get that out of the way. Draw another cell here, which was a smaller cell, and it engulfed this smaller cell. And for whatever reasons, when it engulfed it, it surrounded it, putting out pseudopods probably. Trying to draw the pseudopods which extended. So this was brought into the cell as an organelle. What do they call that in a fusion, whatever. So now we're going to draw this inside the cell. This is the cell membrane of the original cell, and it's surrounded with the organelle membrane. meaning it has two membranes around it. And when we look at the mitochondria and the chloroplast, they do have two membranes around them. They have the original cell membrane, and then they have a second outer membrane, which we call the outer membrane of the mitochondria of the chloroplast. And for whatever reason, this proto-eukaryote did not digest that first cell that it engulfed. It just kept it around, probably to keep it as a reserve food supply. And then when it needed food, it was going to digest that cell. Well, this little cell that got engulfed could perform aerobic respiration, and the proto-eukaryote could not. So the proto-eukaryote only could engage in fermentation, which means 1-glucose could be converted to 2-ATP. Well, this cell could perform aerobic respiration, which means 1-glucose would be converted into 38 ATP. Oops, go back one. And so glucose from the proto-eukaryote went into, oops, need my ruler again. A glucose molecule went from the cytoplasm into the bacteria cell, which was first engulfed. Let me go glucose right here. And probably that just happened by diffusion. That is, the glucose here went there, and the cell wanted the glucose, so it took it up and made it. And then it made 38 ATP. And then it shared some of those ATP with the proto-eukaryote. Why it did this, we may never know. Probably because life was just early, and then some of the ATP dissolved in the water, and then it moved to the cytoplasm, the cell membrane, then moved into the space between the cell membrane and the vesicle membrane, and then moved from the vesicle membrane into the cytoplasm of the proto-eukaryote. How many it shared, we'll never know, but it did share some. And let's just say a lot of ATP, just less than 38 is all we can say. Okay. And the proto-eukaryotes say, wow, what happened here? I lost 1 glucose molecule, but I gained a lot of ATP. This bacteria that I engulfed was going to eat is making me ATP, and it's making me more ATP than I can make from 1 glucose. I'm going to keep this bacteria around and feed it glucose, and then it will make me ATP. And so the two started living in symbiosis, living together. And they lived together now for so long that this bacteria we no longer find free living. We only find it living inside of the cell that engulfed it, which now we're calling a eukaryote, because it has something that we call the mitochondria, which was initially a bacteria free living bacteria that got engulfed by the proto-eukaryote. And then the eukaryote shares glucose with the mitochondria, and then the mitochondria makes ATP and then releases that ATP for use by the eukaryote cell. Any question about any of that? Well, that is how the evolution that the mitochondria happened. At least that's what the theory suggests. Let me close this down now. And the funny thing is that this didn't happen just once. If you look at plant cells, it happened twice. First, they got a mitochondria, which was engulfed by a bacteria that was performing aerobic respiration, became the plant cell's mitochondria, and then they engulfed another cell, which was a photosynthetic prokaryote, and it became the chloroplast of the eukaryote plant cell. And now we don't call it a proto-eukaryote, we're calling it a eukaryote cell because it has mitochondria, and then for plant cells, it has mitochondria and chloroplasts in it. And what evidence do we have to back up this theory? First, if you look at some primitive eukaryotes, which are always protists, you can find that they do engulf other cells. Like this protist, it's a paramecium, engulfed a photosynthetic bacteria, which then is inside the cytoplasm of the paramecium. And then the two live in symbiosis. You can see the bacterium here is actually about to divide. And the paramecium sends nutrients to the bacteria, and then the bacteria can take sunlight and the nutrients and make glucose from CO2. So it can engage in photosynthesis and then share what it makes with the paramecium. We also have algae. There's actually a few species of algae that do not have a mitochondria. And with these algae, not algae, what are they called? Amoebas. What they do, the amoeba cannot engage in aerobic respiration, so they engulf a bacteria that can engage in aerobic respiration, and then the amoeba will live in symbiosis with the bacteria that it engulfed, and it'll send glucose to the bacteria, and then the bacteria will take the glucose, use aerobic respiration, make ATP, and then share that ATP with the amoeba. So that this amoeba that lacks mitochondria is engulfing a bacteria that performs the similar function that the mitochondria does, making ATP for the eukaryote amoeba. So we actually do see this going on in primitive protists. That's the first evidence. The second evidence is if you look at the DNA of a eukaryote cell, the DNA is in linear form. The DNA in a prokaryote cell, like a bacteria, is when one molecule and it's a circular molecule. And we look at the DNA in the mitochondria and the chloroplast, it is one molecule and it's a circular molecule. There are no histones in a bacteria. There's no histones in the mitochondria and the chloroplast, and there are histone proteins on the DNA of a eukaryote cell. If we look at the first amino acid made in a eukaryote cell, it is methionine, meaning AUG codes for methionine. In a bacteria, as well as archaea, you don't need to know archaea, it's a similar amino acid, but a different amino acid. It's formal methionine. So AUG codes for formal methionine. If we look at what AUG codes for in the mitochondria and the chloroplast, it codes for formal methionine. if we look at the ribosomes made from the DNA of the eukaryote cell, meaning the chromosome, they code for a ribosome of 80S in size. If we look at the DNA of a prokaryote chromosome, it makes a ribosome of 70S in size. If we look at the DNA of the mitochondria and the chloroplast DNA, they code for a ribosome of 70S in size, and they make the ribosomes for the mitochondria or the chloroplast. Furthermore, if we look at the growth of eukaryote cells, they grow by mitosis. Prokaryote bacteria mostly grow by binary fission, and we look at the growth of the mitochondria and the chloroplast, and they grow by binary fission. Furthermore, and the strongest piece of evidence that the mitochondria and the chloroplast are bacteria is if we sequence the DNA. If we sequence the DNA of the eukaryote chromosome, the DNA is in the sequence of the domain eukarya. If we sequence the DNA of a bacteria, it will be in the domain bacteria. If we sequence the DNA of the mitochondria and the chloroplast, we find that this DNA sequence is in the domain bacteria. Any question about any of that? All right. That's the endosymbiotic theory, the theory to explain how chloroplasts and mitochondria evolve by being swallowed up by a proto-eukaryote, and then the two go on and live in symbiosis. And nowadays, the mitochondria, as well as the chloroplast, we do not see these bacteria free living anymore on the planet Earth. They're only found inside the cytoplasm of a eukaryote cell. Any question about any of that? All right, that's it for the endosymbiotic theory and for chapter 4. Let's now begin chapter 5. Actually, yeah, let's save that. Chapter 5 is metabolism. Why is that not starting out right? Oh, I got the wrong file. All right, this is correct, where I have the introductory slide showing you the major goals and a rough outline. Always know the terms, the major terms of the chapter. What's catabolism, anabolism? What is activation energy? What's an enzyme? What's an active site? What is competitive inhibition? What is non-competitive inhibition, which is also called allosteric inhibition? What is a redox reaction? What are photoautotrophs? What are photoheterotrophs? What are chemoautotrophs? And what are chemoheterotrophs? You should also know what fermentation is, and then explain the difference between cyclic and non-cyclic photosynthesis. We'll spend a few slides talking about photosynthesis at the very end. Understand what feedback inhibition is, and then lastly, understand what aerobic respiration is. We'll discuss that in great detail. Understand what its steps are and how it differs from anaerobic respiration. Any questions about what we're going to do? All right. You should know, let me blow this up a little, that microbial metabolism is what we're going to talk about. And metabolism is all of the chemical reactions occurring within a cell. And for a bacteria, which we're talking mostly about microbes, bacteria, It is all of the chemical reactions occurring in the organism. You should know that metabolism can be broken down into two parts, catabolism and anabolism. Catabolism is a decomposition reaction where a larger molecule, shrink that down, a larger molecule like starch is broken down into a smaller molecule like glucose, and that releases energy because the larger molecule has more energy than the smaller molecule, or smaller molecules, I guess I should say. Anabolism is a biosynthesis reaction where we take a smaller molecule, such as glucose, and we add several glucose molecules together to make a larger molecule like starch. This requires the input of energy for this reaction to go, so it uses up energy. In your preliminary course, you may have heard of them as an exergonic reaction, releasing energy, that would be the catabolism, and then endergonic reaction, and energy using, where energy must come into the reaction. We're not going to talk about that. We're going to look at the reaction from the size of the molecules, and that's catabolism and anabolism. Let's talk about anabolism because it's a little simpler. Anabolism is a biosynthesis reaction where you take simpler molecules, such as glucose, and it requires the input of energy for this reaction to occur to make a larger molecule, such as starch. Where that energy comes from is usually from ATP. Whenever the cell needs energy to perform its work, what it's trying to do, it usually gets that energy it needs from ATP. So it splits ATP, that releases energy. That energy will be used up to combine the glucose molecules into the starch. And then ATP is broken down into ADP, and inorganic phosphate. Energy is always lost in a chemical reaction. It's known as the second law of thermodynamics. We're not going to talk much about it, but whenever you transfer energy from one molecule to another, some energy is lost as heat. Heat is just another form of energy. So in a sense, the energy isn't lost, it's just converted into heat. Catabolism is where you take a complex molecule and you break it down into a simpler molecule. This gives off energy because the complex molecule stores more energy than the simpler molecule. So energy is given off by this reaction. Whenever energy is converted from one molecule to another, some of that energy is converted into heat. So once again, some of the energy is converted into heat, and that's lost by the system because the system isn't using it, but that's why the term loss comes in. But the energy is just converted from the energy in the molecule to the energy in heat. Sometimes the energy given off can be used to make ATP. So sometimes when we split a complex molecule into a simpler molecule, we can harvest that energy and store some of that energy in ATP. And how you do that is you get ADP and phosphate together, you bring in energy, and then that will fuse the ADP and the phosphate together, forming ATP. This slide is very oversimplified because in animal cells, there are only two reactions that can make ATP. And animal cells have a lot of catabolic reactions, and only 2 of them can make ATP. One of them is where we break down glucose into CO2 and water. That can make ATP. The other is fermentation, where you can, once again, start with glucose, and then you change it to the end products of fermentation, which are mostly lactic acid, depending on which fermentation reaction we're talking about. We're talking about our cells or muscle cells, they make lactic acid, and that can generate ATP. Okay, those are the only 2 ways that animal cells can make ATP. And there are many catabolic reactions in an animal cell, but only 2 of them can make ATP. So that's why I'm saying this slide's a little oversimplified. It's showing that ATP is made in a catabolic reaction, and there's only 2 catabolic reactions that can make ATP. Any question about any of that? Even in plant cells, which they have a few more reactions that can make ATP, there are not too many catabolic reactions that can make ATP. But plant cells do have more than 2. I don't know how many they have, but it's more than 2. But plant cells have a lot of catabolic reactions, and only a few can make ATP. All right. When we're talking about a metabolic pathway, we can have different chemical reactions come together to make a pathway to make the final end product. Let me blow this up so you can see it better. So we have molecule A, which is the starting substrate, and we have an enzyme which can make molecule B, which is the end product of this chemical reaction. But molecule B is the starting product of this reaction happening by enzyme 2 to make molecule C. And then molecule C is the starting product of this reaction by enzyme 3 to make molecule D. And then we have another enzyme, molecule 4, which can take molecule D to make molecule E. And then a 5th enzyme, which takes molecule E to make molecule F. And we can put all of these reactions together in a metabolic pathway, starting with molecule A, and then in the pathway ending with molecule F, which we call the end product because it's the last product of all of these chemical reactions. Molecule A is the starting substrate, also called the reactant, which starts the whole metabolic pathway. Molecules B, C, D, and E are intermediate products. This is a product of this reaction, but it's also the substrate of this reaction, and then etc. for the intermediate products. So we call them that. And then all of these chemical reactions occur by an enzyme. Enzymes are encoded by a gene. So metabolic pathways are determined by genes. Any question about any of that? Up in the show here. Make sure I covered everything there. I did. All right, here are all the chemical reactions that are occurring within a cell. We'll discuss each one of these chemical reactions, and you need to know every single one. Any questions? Any complaints?
No, you're playing, bro. I know you're.
I'm playing with you. You don't
Yeah.
know
Ooh.
the one. That'd be too much for this course. But there's a lot of chemical reactions occurring within a cell, and that slide was trying to show you the different chemical reactions that are occurring within one cell of your body or one cell of a microbe. Chemical reactions occur by the collision theory. The collision theory simply states that a chemical reaction may occur when different atoms, ions, or molecules collide with each other. And if they have enough energy in their collision, then the chemical reaction will proceed. The energy that they need to have the chemical reaction proceed is called the activation energy. The energy needed to disrupt the electronic configurations of the substrates to make the products of that chemical reaction. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes, and then the enzyme will, there's more enzymes, so they will more quickly find a substrate and then convert that substrate to the product. So that increases the reaction rate. You can also increase the temperature. When you increase the temperature, you're making the molecules move around more quickly, so they're more likely to collide. And then when they collide, they're going to have more energy. So you're going to make the reaction happen more quickly just because you've increased the temperature. Pressure, well, I'm not going to explain it, but if you put molecules under pressure, you increase the chance that the reaction will occur because the molecules are under pressure and they will collide with each other and they'll have more energy because the molecules are under pressure. Any question about any of that? Here we're looking at a chemical reaction, and here we have an initial substrate. We can put a y-axis here. That's the energy in the system. Initially, the substrate here has more energy than the products. And so that means energy will be given off by this reaction. How much will be the energy difference from the energy in the starting substrate to the energy in the product? So this will be the energy given off by the reaction. But to go from here to there, you have to go over an energy hump. And that's the energy you need to get the collision to succeed in making the products. That we call the activation energy. And you'll notice there are two activation energy. The activation energy without an enzyme, it's a steep energy hump, and then a reaction enzyme activation energy with an enzyme, and that's less steep. For example, if this were glucose here, I could convert glucose into CO2 and water down here by taking a blowtorch to the glucose and burning it, giving it the activation energy it needs, and that's something like around 1,000 degrees Fahrenheit to convert the glucose into CO2 and water. And yet, glucose is in the room you're in, converted into CO2 and water all the time. And where is that happening? Where is glucose being converted into CO2 and water all the time in the room you're in? Think about that. I'm not going to supply it.
When you're breathing?
I'll give you a hint, though. It's happening at 37 degrees Celsius, which is 98.6 degrees Fahrenheit. And that's when you're breathing inside your cells. Glucose is converted into CO2 and water all the time. And instead of going to 1,000 degrees Fahrenheit, it's only going to 37 degrees, which would be 98.6 degrees Fahrenheit. I don't remember if 1,000 degrees is Fahrenheit or Celsius. I don't remember. what the temperature of a blowtorch is. I know it's around 1,000 degrees, but I don't remember, is that Celsius or Fahrenheit? And it's quite a bit higher than the temperature of your body. That's my point here. Any question about that? So the activation energy without an enzyme is quite a bit higher than the activation energy with an enzyme. Any question about any of that? All right, enzymes are biological catalysts. They speed up a chemical reaction. They speed up a chemical reaction 10 to the 8th to 10 to the 10 times. You don't need to know that number, but the point is they really speed up a chemical reaction quite a bit faster than when without the enzyme around. And for proof of that, if I had a cup of glucose right here, right? Well, over time, that glucose is going to be converted into CO2 and water. But if I were to look at this cup of glucose tomorrow, would I still have a cup of glucose? The answer is yes. If I have a cup of glucose on my table and I look at the cup tomorrow, it's still going to have a cup of glucose. It's going to take a long time to convert a cup of glucose into CO2 and water. It will eventually happen. Whenever a cosmic ray particle hits the glucose, it will split the glucose into CO2 and water. But that's going to take centuries to convert a cup of glucose into CO2 and water. And yet, if I were to eat that cup of glucose, I don't know how soon it would be digested into CO2 and water, but it would eventually, well, certainly within a day, be converted into CO2 and water, unless I were not being very active, in which case the glucose would be converted into fat. The point is, it's converted. Any questions about any of that? Enzymes are specific for a chemical reaction. Their specificity is due to their three-dimensional shape. Each enzyme is specific for one specific substrate, and then that enzyme will change that one specific substrate to its products. That's not exactly true. As you know, there are some drugs that mimic the substrate, and then the drug can combine with the enzyme and tie up the enzyme. But for our purposes, we're going to talk in simple terms, and that is one enzyme works specifically on one specific substrate. As I stated, that's not entirely true, but for our purposes, it's true enough. And then the enzyme will convert that substrate into the products, and then the enzyme is not used up by the chemical reaction, and that enzyme can then go bind to another substrate and convert that other substrate into the products, and that can continue on and on again. The point is the enzyme is not altered in the reaction, and it's not used up. An example of something that gets used up, it's not an enzyme, but when you have trying to remember the name of that molecule, a free radical in your body, you can combine vitamin C with it. The vitamin C will combine with the free radical and then tie it up and then get rid of the free radical. That also gets rid of the vitamin C. It's no longer vitamin C that's useful for you. And I don't know what happens to it. You probably pee it out because you get rid of the free radical that way too. But the point is that vitamin C gets used up by by complexing with a free radical and then removing the free radical. The enzyme is not used up. Enzymes, when they bind to their substrate, they actually form a complex with the substrate. So the enzyme forms a substrate complex, meaning the enzyme substrate is a complex, and they're briefly bound together. This actually orients the substrate, increasing the probability that the reaction will occur. The complex substrate puts stresses on the chemical bonds in the substrate to help convert those bonds into the bonds of the product. And that could be breaking the bonds of the substrate to make the product. And that lowers the activation energy. Any question about any of that? We'll talk a little bit more about the substrate in just a few minutes. The enzyme substrate complex, in just a few minutes. First, I want to mention that enzyme names usually end in ASE. You don't need to know the examples, but we will talk about catalase, ligase, polymerase, and those enzymes all end in ASE. That's not all enzymes end in ASE, like renin is an enzyme in our stomach to curdle milk, and it's an important enzyme for babies because when the baby curdles the milk is in the stomach for a longer period of time, and that allows the stomach to digest more of the milk. The point is, renin is an enzyme. It doesn't end with ACE, but essentially all enzymes that were named after this convention end in the name ACE. Renin is an enzyme that was known in ancient times, I think Roman times or maybe Greek times, something like that. And that's why it wasn't ending in ACE, because when they named it, that convention wasn't in force yet. You should also know that enzymes do not work in a vacuum. Some enzymes have a helper, either a coenzyme or a cofactor, that binds to the enzyme, which then allows the enzyme to bind to the substrate. You don't need to know the names of the enzyme without the helper and the enzyme with the helper. You should know that there are two helpers. A coenzyme, which is a biological molecule, like NAD or FAD, is a coenzyme. A cofactor is usually a chemical element like iron, zinc, magnesium. And both of them just bind to the enzyme. And then the enzyme and its helper can then bind to the substrate, so that the active site, this part right here, actually binds to the substrate. Some important coenzymes, you should know the first two. NAD plus is a coenzyme we're going to talk about in aerobic respiration, and NAD plus stands for nicotinamide adenine dinucleotide. It's primarily involved in catabolic reactions. We'll talk about it with aerobic respiration. FAD, flavine adenine dinucleotide, also involved in catabolic reactions. We'll talk about it in aerobic respiration as well. The other reaction coenzymes we're not going to talk about much. And so you just need to really know the first two as coenzymes. How an enzyme works is it binds to a substrate. Let me blow this up. And it has an active site of the enzyme right here. that actually binds to the substrate, and the specificity of the enzyme for one substrate is dependent on the shape of the active site. And so the molecule has to be in its correct three-dimensional form for the enzyme to have the correct active site. And then that active site can bind to the substrate. And that's pretty much why one enzyme binds to one substrate, because it actually does fit that substrate, binds to it, forms a complex between the enzyme and the substrate that puts stresses on this substrate, which then makes the chemical bond break right here. And once that happens, once that happens, the substrate is now converted into the products. The products fall off of the enzyme, freeing the enzyme to then go bind to more substrate. This model shown here is the lock and the key model, where a specific key fits a specific lock. I'm not sure which one's supposed to be the key and which one's supposed to be the lock. This might be the key and not the lock, or I don't know, it doesn't matter. The point is there's one key to fit one lock, and then that key can open up the lock. And that's the key and lock model of an enzyme. Any question about that? Here we're looking at the hand in the glove model, where the enzyme changes its shape to fit the substrate, much like a glove changes its shape to fit the hand. And that's why it's called the hand in the glove model of an enzyme. What do you notice by this model? There's three things you might notice. One's pretty obvious. Looking at that, what can you state? Come on. I want a little bit of...
The enzyme wraps around the substrate?
Yeah, the enzyme changes its shape to fit the substrate. So the enzyme changes its shape to fit the substrate. Now that's the first thing. What do you notice about the size of the enzyme and the substrate? That's the very easy one. Which one's bigger?
The enzyme is very big compared to the substrate?
Yeah, the enzyme is much bigger than the substrate, and that's generally true for enzymes. They're much bigger than the substrate. So that's the second thing you should notice. The third thing you have to look at very closely, and I'll blow it up so it's a little more apparent, and that is, you see the substrate shape right here, and then the substrate shape right there. The substrate actually changes its shape to fit the enzyme. It's a little flatter here than it is there. And that you could call the shapeshifter's hand. And the shapeshifter's hand fitting the glove, where the glove is actually molding to fit the hand, and then the hand is molding to fit the shape. And those of you who know Star Trek Deep Space Nine know what I'm talking about. When I'm talking about the shapeshifter's hand, Udo's hand could change its shape to fit the glove. Okay? And that's actually what happens when an enzyme and a substrate complex. Both the enzyme changes its shape to fit the substrate, and the substrate changes its shape to fit the enzyme. All right. And then the enzyme, once it's done, the product comes off, and that enzyme can then repeat this reaction 1,000 times to 10,000 times per second, meaning it can do this many, many times. speeding up the chemical reaction. There are different factors that influence an enzyme. An enzyme does not work in a vacuum. The temperature, the pH, the salinity, the presence of heavy metals, the presence of alcohol, the presence of radiation, UV radiation, can cause the protein of the enzyme to denature, in which case the enzyme will have less activity. And if the protein is totally denatured, the enzyme will have no activity. So temperature, pH, salinity, presence of heavy metals, presence of alcohol, the presence of UV radiation can influence the enzyme's activity. The substrate concentration can also influence the enzyme's activity. When there's no substrate around, the enzyme's not going to have any activity. You increase the substrate concentration a little bit, you increase the enzyme's activity. You increase the substrate concentration further, you increase the enzyme activity. We'll talk a little bit about that. Generally, as you increase the substrate concentration, you increase the enzyme activity up into a point where then it plateaus and you increase the substrate concentration. It does no longer increase the enzyme activity. And we'll talk about that in a little bit. There are different poisons which can be around. Poisons to enzymes like cyanide or fluoride. They can influence the enzyme activity. They destroy the enzyme. And obviously, if there's only one enzyme around, one molecule of an enzyme around, and you have a poison around, well, you have no longer any functional enzyme. But if you had two enzymes around and one molecule of cyanide, it gets together with the enzyme, and now you only have one functional enzyme and one non-functional enzyme. The point is, enzymes can influence the activity of an enzyme. You can also have inhibition influence the enzyme activity. There's two types of inhibition: competitive inhibition and non-competitive inhibition, also called allosteric inhibition. And then we'll talk briefly about feedback inhibition. So here is a functional active enzyme in its three-dimensional shape. You can denature this enzyme where the enzyme no longer has its three-dimensional shape, and it takes on this shape by the incorrect temperature, the incorrect pH, incorrect salinity. And this looks like the enzyme's totally denatured, so the enzyme will have no activity. So enzymes don't work in a vacuum. Different temperature, different pH do affect the amount of enzyme activity. For this enzyme, it works best at temperature 37. When you go away from 37, the amount of enzyme activity decreases. When you go way away from 37, the enzyme activity decreases until there's no activity left in the enzyme. So if you heat up the protein beyond 50 degrees, the enzyme will have no activity and the protein will be totally denatured. Any question about any of that? The same thing if you lower the temperature. You get it too far away from its optimal temperature, the enzyme will have no activity. Same thing with pH. This one has an optimal pH, the enzyme around pH 5. And when you move away from that pH, the enzyme activity decreases even further. When you move way, way away from that pH, like around pH 0 or pH 10, the enzyme is totally denatured, has no activity. Where would we have an enzyme that would work around pH 5? Anyone from A and B know that question? Where do I have an enzyme around that works best around pH 5? Well, I'll give you a hint. It's not your stomach because that's around pH 1 to pH 2. And this enzyme works best around pH 5. It has very little activity around pH 1. Your skin is around pH 5. So this enzyme is probably an enzyme in your skin cell. Your blood would be working best around pH 7 because that's the pH of your blood. So this would not be an enzyme in your blood. For the enzyme concentration, when we have no substrate concentration, there is no enzyme activity, and that's not surprising because there's no substrate for the enzyme to work on. And we increase the substrate concentration, we get more and more enzyme activity because the enzyme is more quickly able to find the substrate and then convert it into the product. And as we increase the substrate concentration, the rate starts to decrease, and eventually we reach a plateau where when we increase the substrate concentration further, there is no increase in the enzyme activity. Anyone have a guess for why that might be? It's actually pretty simple. Here, we have enough substrate in solution, but all of the enzyme is currently bound to the substrate. So all of the enzyme is currently bound to the substrate. So if we increase the substrate concentration further, you cannot speed up the enzyme activity any because all of the enzyme is already bound. So you can't speed it up. Any question about any of that? All right, let's talk about inhibition. An inhibitor can compete with the substrate for the reactive site, and this we call a competitive inhibitor, one that actually binds to the active site, and now the substrate cannot bind to the enzyme. We have certain drugs that work this way, where they will bind to an enzyme, and then the enzyme cannot act on the substrate to make the products. And that is competitive inhibition, where the inhibitor is competing for the active site with the substrate. Any question about any of that? We also have non-competitive inhibition or allosteric inhibition, where the inhibitor binds to the enzyme, but it does not bind in the active site of the enzyme. Here, the non-competitive inhibitor or allosteric inhibitor binds in an allosteric site, a site other than the active site. And when the inhibitor binds to this enzyme, it changes the folding pattern of the enzyme, changing the shape of the active site. So now the active site could no longer bind to the substrate, and that will inhibit this enzyme. Any question about that? All right. Non-competitive inhibition, where the inhibitor binds at an allosteric site, a site other than the active site, changing the three-dimensional folding pattern of the enzyme so that the enzyme can no longer bind to the substrate. We can link the, let me get this over there a little. Feedback inhibition is where you have a reaction starting with a substrate, which is converted by the first enzyme to an intermediate, converted by another enzyme to another intermediate, converted by another enzyme to the end product. So we have a reaction pathway to make the end product from the substrate. Feedback inhibition is where the end product itself can bind to the first enzyme as an allosteric inhibitor, changing the shape of the active site so that the enzyme can no longer bind to the substrate. So when the substrate or the end product is low in concentration in the cell, we have the substrate making intermediate A, which is made into intermediate B, which is made into the end product, and that makes more end product. As the concentration of the end product increases in the cell, more and more of the end product will bind to the enzyme, shutting down the enzyme and feedback inhibition until you get so much end product in the cell that the end product binds to every enzyme, and that totally shuts down the production of the end product. Okay, no more end product is being made by feedback inhibition. And then the end product, let's just say, is slowly degraded in the cell. Eventually, the end product will fall off of the enzyme because it's binding by a stoichiomic reaction. So every once in a while, it falls off of the enzyme. When there's a lot of end product around, another one will just bind to the enzyme and inactivate it. But if there's not a lot of end product around, there's nothing to bind to the enzyme. And as more and more enzyme falls off, that will activate more and more enzyme 1. We'll get more and more intermediate A made, more and more intermediate B made, and that will resume production of the end product. And this is the way feedback inhibition occurs in a cell. Feedback inhibition almost always is this way, where the end product itself shuts down the first enzyme. And the reason why we see this is if you were to shut down a second enzyme, like enzyme 2 or enzyme 3, you're going to be making a product. And that costs the cell energy. And so you may as well make this system as efficient as you can. And the most efficient is to shut down the first enzyme by the end product itself. And then when you don't have enough end product in the cell, it starts falling off the enzyme, and that will reactivate production of the end product. And that's why we see feedback inhibition in this way in cells. Any question about any of that? All right, there's no question. Let's move on. Let me see at time. I'm having a hard time now. We got a little bit of time. We need to talk about ribozymes. Ribozymes are non-protein enzymes. Most enzymes are protein, but there are a few enzymes which are made of RNA. So ribozymes are RNA enzymes. They are RNA molecules that cut and splice RNA or other molecules. They do function as an enzyme in that these RNA enzymes have an active site that binds to the substrates. These RNA enzymes are not used up or modified in the chemical reaction. So once the product is made, this ribozyme is then free to bind to more substrates to perform more chemical reactions. A lot of ribozymes actually are involved in cutting and splicing RNA, but not all of them. The only ribozyme we'll ever mention in the class is when the ribosome in aerobic respiration takes the growing polypeptide off of the first tRNA and then combines it to the second amino acid on the second tRNA, that is actually happening by an RNA enzyme. And that's the only RNA enzyme we will discuss in the class. And I'm only mentioning it to you because this is one enzyme we'll talk about. And your textbook author does not mention it, but this is the only RNA enzyme we'll mention in the class. And we'll discuss that when we discuss aerobic respiration, which will be later in this lesson. Any question about any of that? All right, if not, let's move on to a different topic, metabolic diversity among organisms. All organisms, including microbes, require an energy source and a carbon source. And we can separate all living things. We're going to only do it with microbes, but we can separate all microbes into what their energy source is and what their carbon source is. Any question about any of that? So, diversity among organisms, we can classify them on where they get their energy source. They are a phototroph if they derive their energy from light. They are a chemotroph if they derive their energy from an inorganic or an organic chemical compound. What are we, a phototroph or a chemotroph?
Chemotroph.
We're a chemotroph. And then everybody knows green plants are phototrophs. You can also classify organisms on where they get their carbon source. Organisms are autotrophs if their principal carbon source comes from CO2. CO2 either in the air or in the water. Heterotrophs use an organic source for their carbon. So we get our carbon source from mainly glucose, but any food product we eat, we can get organic carbon source from the food we eat, such as glucose. But green plants get their primary carbon source from CO2. So green plants are an autotroph and we are a heterotroph. If ever you get confused on the organism and what its troph is, just broke it down into where does it get its energy source. If it gets it from light, it's a phototroph. If it gets it from a chemical, it's a chemotroph. And then break it down by its carbon source. Where does it get its carbon? If it gets it from CO2, it's an autotroph. If it gets it from an organic chemical for its carbon, then it is a heterotroph. We can combine all of these four together, in which case it's a little more complicated, where we classify the organism both on where it gets its energy source and its carbon source from. There are four classifications: a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. Photoautotrophs is probably the easiest, so I'll talk about that one. Well, I guess the chemoheterotroph is also easy. Photoautotroph is a little easier, and that is it's a phototroph, so it gets its energy needs from light, and it's an autotroph, so it gets its carbon needs from CO2. Green plants are a photoautotroph, but so are green photosynthetic bacteria like the cyanobacteria. We'll talk a little bit more about the oxygenic and the non-oxygenic photoautotrophs later. I'm not going to talk about it now. Too complex. The next simplest is probably the chemoheterotrophs. They get their energy source from a chemical compound, such as glucose, and they get their carbon source from an organic chemical compound, such as glucose. Oftentimes, the same molecule that gives the carbon source, gives the energy source, such as glucose, which is both a chemical energy source for us and a chemical carbon source for us. And the animals all fall in this chemoheterotroph, as well as the fermentative bacteria, bacteria like E. coli, animals, most of the protozoa, all of the fungi, and many bacteria are chemoheterotrophs. What's complicated about the chemoheterotrophs is that their energy source can come from an organic chemical molecule such as glucose, or it can come from an inorganic chemical molecule such as nitrate or nitrite or sulfate or iron. And we'll talk a little bit about those after I talk about the chemoautotrophs. The chemoautotrophs get their chemical source from, their energy source from a chemical, and it's an inorganic chemical, such as nitrate, nitrite, sulfur, iron, hydrogen sulfide. And they get their carbon source from CO2. The chemosynthetic bacteria can be a chemoautotroph. If the chemosynthetic bacteria is getting its energy source from an organic chemical compound, then that is a chemoautotroph, chemosynthetic bacteria. However, the chemosynthetic bacteria can be chemoautotrophs or they can be chemoheterotrophs. If the chemosynthetic bacteria gets its carbon from an organic chemical compound, such as glucose, this is a chemosynthetic bacteria, which is a chemosynthetic bacteria that's a chemoheterotroph, as to opposed to a chemoautotroph. And this chemoheterotroph can get its energy source from an inorganic chemical compound, such as iron, or nitrate, or nitrite, or sulfate. And that's very different from us, when we can only get our energy needs from an organic chemical compound, such as glucose. Any question about that? All right. The complicated part is the chemosynthetic bacteria can be either a chemoautotroph or a chemoheterotroph, depending on where they get their carbon source from. You actually know a chemosynthetic bacteria, which is a chemoautotroph. And that is if you lift up the toilet tank, not the toilet bowl, but the tank that stores the water for your toilet, you'll see some brown or yellowish slime on that toilet tank. That is largely iron oxidizing bacteria, getting the iron from the water, being its energy source, and then it's getting CO2 from the air dissolved in the water for its carbon source. And so I'm sure all of you have seen this iron oxidizing chemoautotroph bacteria. Any question about any of that? All right, the most difficult one to know is the photoheterotrophs. There are phototrophs, so they get their energy needs from light, but they get their carbon source from an organic compound, such as glucose. The green and the purple non-sulfur bacteria are an example of a photoheterotroph. And how many of you have ever heard of the green and purple non-sulfur bacteria? Anyone? Probably not. Okay, any questions about the different trophs? All right, let's talk a little bit more about the photoautotrophs. You can have two types of photoautotrophs. There can be oxygenic, and they produce oxygen in photosynthesis. And there can be anoxygenic. They do not produce oxygen in photosynthesis. You don't need to know the examples, but obviously cyanobacteria and green plants are oxygenic. And you probably don't know the anoxygenic photosynthetic organisms, but the green and purple sulfur bacteria are photoautotrophs, but they do not produce oxygen. In the next slide, you should know everything in red. We can separate all organisms by their energy source. If they get their energy needs from light, they're a phototroph. And then we can separate the phototrophs by where they get their carbon source. If they get their carbon from CO2, they are a photoautotroph. If they get their carbon source from an organic chemical compound, such as glucose, they are a photoheterotroph. And you don't need to know the examples. And we're not going to test you on oxygenic photosynthesis and anoxygenic photosynthesis. We're not going to test you on that. But you should know that if the organism gets its energy source from a chemical compound, they are a chemotroph. And this could be a chemical compound if they're chemosynthetic bacteria, or it can be an organic compound if they are not a chemosynthetic bacteria, meaning another form of bacteria that's a chemotroph, as well as animals are chemotrophs. You can then split the chemotrophs by where they get their carbon source. If they get their carbon from CO2, they are a chemoautotroph. If they get their carbon source from an organic chemical compound, they are a chemoheterotroph. Animals are chemoheterotrophs. Some bacteria are chemoautotrophs, including some of the chemosynthetic bacteria are chemoautotrophs, but some of the chemosynthetic bacteria are chemoheterotrophs. Okay. Any question about that? We're not going to talk about the final electron acceptor. Can split those two. All right. So that's the classification of organisms. We're very close to ending. I think I'll stop here because this is a good place to stop. Oh, good heavens, it's actually time to stop. I thought it was before time to stop. All right. Any questions before I go? If not, I'll see you in the lab at a little after 7. I've got something to do, so I might be a minute or two late.